Radiopharmaceuticals have proven to be effective agents since they can be successfully applied for diagnostics and therapy. Traditional radiotherapy is administered from outside the body, without differentiating between normal and cancerous tumor cells. Radionuclide drug conjugates combine a radioactive compound that emits radiation, a tailored targeting molecule for specific receptors enriched on the surface of diseased cells, and a linker for stable attachment, enabling more effective radiation delivery and significantly reducing side effects by attacking only cancerous cells. Since 2016, 11 RDC drugs have been approved worldwide, of which 9 have been approved by the FDA, including 7 for cancer diagnoses and 2 for cancer therapy. RDCs are made up of 4 major components: radionuclide, chelator, linker, and ligand. Radionuclide, the biggest difference between RDC and other conjugate drugs, is the payload. RDC payloads are no longer nonspecific toxins, but radionuclides. This is the radioactive atom that emits radiation, capable of killing a cancerous cell.
The choice of radionuclide deployed may depend on whether the chosen application is to visualize enrichment in the area of high concentrations of the target receptor, in support of a diagnosis or targeting therapeutic strategies, or to support the cell-killing delivery of a toxic radionuclide. The huge advantage of this type of molecule is the potential to insert different metal atom radionuclides that can be used for different purposes in different settings. However, the same radiopharmaceutical can often accommodate a cell-killing radiometal, such as an alternative isotopic form of the same metal used for diagnosis, or alternative radiometals such as lutetium-177 or actinium-225.
Targeting ligand. Like a homing beacon, this molecule locates and binds to the surface of diseased cells. Targeting molecules can be a peptide, an antibody, or another substance that has a high affinity and selectivity for a receptor on a diseased cell. Linker. The linker plays a crucial role in the safety and stability of the RDC. Non-cleavable linkers are normally used by RDCs for safety considerations. Chelator. The chelator binds the radionuclide, ensuring it's securely in place and can be delivered to the diseased cells without affecting healthy tissue.
The diversity of chelating capability is illustrated with a number of very well characterized cyclic nitrogen-containing ring structures able to chelate different radiometals. The convenience here is that the insertion of the radiometal is invariably the final step in the synthesis of the radioimaging or radiotherapy molecule. So the cold parent molecule may be prepared on scale and transported to the intended site of use, such as a hospital, for the final step of insertion of short half-life radiometals.
WuXi AppTec has developed an optimized workflow for radiopharmaceutical discovery and development, which encompasses the selection of a target, identification of an appropriate ligand for the target, incorporation of a chelating group, assessment of target affinity, and measurement of the ligand's metabolic stability. It further involves the evaluation of diverse strategies for radionuclide incorporation, in vivo assessment for efficacy, distribution, and metabolism, as well as toxicology, CMC, and regulatory submissions.